Hi, myself Dr. Vijay Misra, Associate Professor, Edina Institute of Pharmaceutical Sciences, Sagar, MP. I am here with an interesting and learning episode of B.Pharma on the important unit of drug interactions, including drug-drug interaction with reference to analgesics, anti-inflammatory drugs, diuretics, and antibiotics, which is one of the important units of section drug design of B.Pharma 8th semester. Let's start our episode while taking a look on what we are going to learn today. Module 1st, drug-drug interactions with reference to analgesics. Module 2nd, drug-drug interaction with reference to anti-inflammatory drugs. Module 3rd, drug-drug interactions with reference to diuretics. Module 4th, drug-drug interactions with reference to antibiotics. Let's discuss about drug-drug interaction. Then we will move to drug-drug interaction with reference to analgesics. An interaction can either increase or decrease the effectiveness and or the side effects of a drug, or it can create a new side effect. The drug interactions can reduce the desired effects of a drug, increase the adverse effect of a drug, or result in unnecessary pain and suffering. However, on the other hand, drug interaction may increase the beneficial effects of a drug and can decrease the adverse effects of a drug. Now, students, we will move to the module 1st, drug-drug interaction with reference to analgesics. Algesia, that is pain, is an ill-defined, unpleasant bodily sensation, usually evoked by an external or internal noxious stimulus. Analgesic is a drug that selectively relieves pain by acting on the CNS, that is central nervous system, or on peripheral pain mechanism without significantly altering consciousness. Pain is a warning signal, primarily protective in nature, but causes discomfort and suffering, may even be unbearable and incapacitating. It is the most important symptom that brings the patient to the physician. Analgesics relieve pain as a symptom without affecting its cause. They are used when the noxious stimulus evoking the pain cannot be removed or as an adjuvant to more etiological approach to pain. Analgesics are divided into two groups. First one is opioid, or it is also known as narcotic or morphine-like analgesics. Second is non-opioid. It is also known as non-narcotic or aspirin-like antipyretic or anti-inflammatory analgesics. Now, opioid analgesics. These can be classified as first, natural opium alkaloid. Example of this are morphine and codeine. Second is semi-synthetic opiates. Example is diacetylmorphine, that is also known as heroin, phenylcodeine, ethylmorphine. Many others like hydromorphone, oxymorphone, hydrocodone, oxycodone are not used in India. Third one is synthetic opioids. Examples are pethidine, fentanyl, methadone, tramadol. Many others like levorphenol, dextromoramide, afentanil, sufentanil, remifentanil are not available in India. Chemically, codeine is methyl morphine, occurs naturally in opium and is partly converted in the body to morphine. It is less potent than morphine, that is 1/10th as analgesic. The degree of analgesia is comparable to aspirin, that is 60 mg of codeine is equivalent to 600 mg aspirin and can relieve mild to moderate pain only. Various drugs like phenothiazine, tricyclic antidepressant, monoamine oxidase, that is MAO inhibitors, amphetamine and neostigmine potentiate the action of morphine and other opioids, either by retarding its metabolism or by a pharmacodynamic interaction at the level of central neurotransmitters. Morphine retards absorption of many orally administered drugs by delaying gastric emptying. Now second is non-opioid analgesics. All drugs grouped in this class are also called non-narcotic, non-opioid or aspirin-like analgesics. These have analgesic, antipyretic and anti-inflammatory actions in different measures. In contrast to morphine, they do not depress central nervous system, that is CNS, do not produce physical dependence, have no abuse liability and are weaker analgesics except for inflammatory pain. They act primarily on peripheral pain mechanism but also in the CNS to raise pain threshold. They are more commonly employed and many are over the counter, that is OTC drugs. This class of analgesics shows various drug-drug interactions. Some of them are as follows. First one, aspirin displaces warfarin, naproxen, sulfonylureas, phenytoin and methotrexate from binding sites on plasma proteins, so toxicity of these drugs may occur. Its antiplatelet action increases the risk of bleeding in patients on oral anticoagulants. Second, aspirin at analgesic doses inhibits tubular secretion of uric acid and antagonizes uricosuric action of probenecid. Tubular secretion of methotrexate is also interfered. Third one is aspirin blunts diuretic action of furosemide and thiazides and reduces potassium ions conserving action of spironolactone. Competition between careninone, active metabolite of spironolactone, and aspirin for active transport in proximal tubules has been demonstrated. Fourth, aspirin reduces protein-bound iodine levels by displacement of thyroxine, but hypothyroidism does not occur. Next drug, ibuprofen, was the first member of propionic acid derivatives introduced in 1969 as a better tolerated alternative to aspirin. Another propionic acid derivative, phenacetin, introduced in 1887 was extensively used as analgesic antipyretic, but is now banned because it was implicated in analgesic abuse nephropathy. Paracetamol, acetaminophen, is deethylated active metabolite of phenacetin. All the propionic acid derivatives are well absorbed orally, highly bound to plasma proteins, that is 90 to 99%, but displacement interactions are not clinically significant. The dose of oral anticoagulants and oral hypoglycemics need not to be altered because they inhibit platelet function. Use with anticoagulants should nevertheless be avoided. Similar to other NSAID, that is non-steroidal anti-inflammatory drugs, they are likely to decrease diuretic and antihypertensive action of thiazides, furosemide and beta blockers. The cause and nature of pain, that is mild, moderate or severe, acute or chronic, ratio of pain, inflammation, along with consideration of risk factor in the given patient, that is age, concurrent disease and drug therapy, history of allergy, govern selection of analgesics. Also to be considered are the past experience of the patient, acceptability and individual preference. Patients differ in their analgesic response to different NSAID. If one NSAID is unsatisfactory in a patient, it does not mean that other NSAID will also be unsatisfactory. Some subjects feel better on a particular drug, but not on a closely related one. It is in this context that availability of such a wide range of NSAID may be welcome. Module 2. Drug-drug interaction with reference to anti-inflammatory drugs. The most important mechanism of anti-inflammatory action of NSAID is considered to be inhibition of cyclooxygenase 2, that is COX-2 mediated enhanced prostaglandins, that is PG synthesis at the site of injury. However, there is some evidence that inhibition of the constitutive COX-1 also contributes to suppression of inflammation, especially in the initial stages. The anti-inflammatory potency of different compounds roughly corresponds with their potency to inhibit COX. However, nimesulide is a potent anti-inflammatory but relatively weak COX inhibitor. PG, that is prostaglandins, are only one of the mediators of inflammation. Inhibition of COX does not depress the production of other mediators like LTs, PAF, cytokinase, etc. Inflammation is the result of concerted participation of a large number of vasoactive, chemotactic and proliferative factors at different stages, and there are many targets for anti-inflammatory action. Activated endothelial cells express adhesion molecules, that is ELAM1, ICAM1 on their surface and play a key role in the directing circulating leukocytes on the site of inflammation, that is chemotaxis. Similarly, inflammatory cells express selectins and integrins. Certain NSAID may act by additional mechanisms including inhibition of expression, activity of some of these molecules and generation of superoxide, other free radicals, growth factors like GM, CSF, IL-6, that is interleukin, as well as lymphocyte transformation factors and TNF alpha may also be affected. Stabilization of leukocyte, lysosomal membrane and antagonism of certain actions of kinase may be contributing to NSAID action. Dear student, in 1971, 1A and co-workers made the landmark observation that aspirin and some NSAID blocks PG generation. This is now considered to be the major mechanism of action of NSAIDs. Prostaglandins, prostacyclin, PG12 and thromboxane A2, that is TXA2, are produced from arachidinoic acid by the enzyme cyclooxygenase, which exists in a constitutive COX-1 and an inducible COX-2 isoforms. The former serves physiological housekeeping functions, while the latter, normally present in minute quantities, is induced by cytokinins and other signal molecules at the site of inflammation. Generation of PG locally, which mediates many of the inflammatory changes. However, COX-2 is constitutively present at some sites in brain, in juxtaglomerular cells and in the fetus. It may serve physiological role at these sites. Most NSAID inhibit COX-1 and COX-2 non-selectively, but now some selective COX-2 inhibitors have been produced. Features of non-selective COX-1, COX-2 inhibitors, that is traditional NSAIDs, and selective COX-2 inhibitors are compared in table. Aspirin inhibits COX irreversibly by acetylating one of its serine residues. Return of COX activity depends on synthesis of fresh enzyme. Other NSAID are competitive and reversible inhibitors of COX. Return of activity depends on their dissociation from the enzyme, which in turn is governed by the pharmacokinetic characteristics of the compound. Aspirin is acetyl salicylic acid. It is rapidly converted in the body to salicylic acid, which is responsible for most of the actions. Other actions are the result of acetylation of certain macromolecules including COX. It is one of the oldest analgesic anti-inflammatory drugs and is still frequently used. Anti-inflammatory action is exerted at high doses, that is 3 to 6 gram per day or 100 mg per kg per day. Signs of inflammation like pain, tenderness, swelling, vasodilation and leukocyte infiltration are suppressed. In addition to COX inhibition, quenching of free radicals may contribute to its anti-inflammatory action. Aspirin is rapidly deacetylated in the gut wall, liver, plasma and other tissues to release salicylic acid, which is the major circulating and active form. It is about 80% bound to plasma proteins and has a volume of distribution about 0.17 liter per kg. Aspirin therapy in children with rheumatoid arthritis has been found to raise serum transaminases, indicating liver damage. Due to risk of Reye syndrome, pediatric formulations of aspirin are prohibited in India and the UK. All propionic acid derivatives inhibit PG synthesis, that is prostaglandin synthesis. Naproxen being the most potent, but their in vitro potency to inhibit COX does not closely parallel in vivo anti-inflammatory potency. Inhibition of platelet aggregation is short lasting with ibuprofen, but longer lasting with naproxen. All are well absorbed orally, highly bound to plasma proteins, that is 90 to 99%, but displacement interactions are not clinically significant. Those of oral anticoagulants and oral hypoglycemics need not to be altered because they inhibit platelet function. Use with anticoagulants should nevertheless be avoided. Similar to other NSAID, they are likely to decrease diuretic and antihypertensive action of thiazides, furosemide and beta blockers. Dear friends, till now you have gained a complete idea about drug-drug interaction with reference to analgesics, anti-inflammatory drugs. If you have missed any of the module or content, you may log on to our website, that is www.cec.nic.in to download videos, FAQ, LOR and other contents so that you can remain updated with this chapter. Now we move ahead to our next modules that are about drug-drug interaction with reference to diuretics and antibiotics. Module 3rd, drug-drug interaction with reference to diuretics. Diuretics, that is natriuretics, are drugs which cause a net loss of sodium ion and water in urine. However, sodium ion balance is soon restored even with continuing diuretic action by compensatory homeostatic mechanism of the body, albeit with a certain degree of persisting sodium ion deficit and reduction in extracellular fluid volume. Based on the diuretic action of calomel, an organomercurial given by injection were introduced in the 1920s and dominated for nearly 40 years. The carbonic anhydrase inhibitors were developed in the 1950s from the observation that early sulfonamides caused acidosis and mild diuresis. The first modern orally active diuretic, chlorothiazide, was produced in 1957 and by early 1960 its congeners like thiazide diuretics were already in common use. Availability of furosemide and ethacrynic acid by mid-1960s revolutionized the pattern of diuretic use. The aldosterone antagonist and other potassium-sparing diuretics like spironolactone and triamytrin amiloride were developed in parallel to these. The diuretics can be classified as follows. First one, high ceiling or loop diuretics. Example are furosemide, bumetanide, toracimide. Thiazide and related diuretics. Example are chlorothiazide. Next is carbonic anhydrase inhibitors. Example is acetazolamide. Next is potassium sparing diuretics. Examples are spironolactone, triamytrin, amiloride. Next is osmotic diuretics. Examples are mannitol, isosorbide, glycerol. Furosemide has weak carbonic anhydrase inhibitory action. Furosemide is rapidly absorbed orally, but bioavailability is about 60%. Bumetanide, it is similar to furosemide in all respects, but it 40 times more potent. It induces very rapid diuresis. Toracimide or torcimide, another high ceiling diuretic with properties similar to furosemide but 3 times more potent. All thiazides and related drugs are well absorbed orally. There are no injectable preparations of these drugs. Their action starts within 1 hour, but the duration varies from 6 to 48 hours. Potassium sparing diuretics are more efficaciously and more convenient in correcting hypokalemia than are potassium ion supplements. ACE inhibitors and AT1 antagonists given with thiazides tend to prevent development of hypokalemia. Other drug-drug interactions in relation to diuretics are as follows. First one, thiazides and high ceiling diuretics potentiate all other antihypertensives. This interaction is intentionally employed in therapeutics. Second is hypokalemia induced by these diuretics enhances digitalis toxicity, increases risk of polymorphic ventricular tachycardia. Third one is high ceiling diuretics and aminoglycoside antibiotics are both ototoxic and nephrotoxic, produce additive toxicity, should be used together cautiously. Fourth one is cotrimoxazole given with diuretics has caused higher incidence of thrombocytopenia. Fifth one, indomethacin and other NSAID diminish the action of high ceiling diuretics by inhibiting Pg synthesis in the kidney, through which furosemide and related drugs induce intrarenal hemodynamic changes which secondarily affect salt output. Antihypertensive action of thiazide and furosemide is also diminished by NSAID. Sixth one is probenecid competitively inhibits tubular secretion of furosemide and thiazide, decreases their action by lowering concentration in the tubular fluid, while diuretics diminish uricosuric action of probenecid. Seventh is serum lithium level rises due to enhanced reabsorption of lithium ion and sodium ion in proximal tubule. Potassium sparing diuretics, aldosterone antagonist and renal epithelial sodium ion channel inhibitors indirectly conserve potassium ion while inducing mild natriuresis and are called potassium sparing diuretics. When given together with potassium ion supplements, dangerous hypokalemia can occur. Second, aspirin blocks spironolactone action by inhibiting tubular secretion of its active metabolite canrenone. Third one is more pronounced hypokalemia can occur in patients receiving ACE inhibitors and ARBs. Fourth one is spironolactone increases plasma digoxin concentration. Spironolactone interacts with progestin and androgen receptors as well. In addition, it may enhance testosterone clearance or its peripheral conversion to estradiol, reducing dose and duration of treatment related hormonal side effects like gynecomastia, erectile dysfunction or loss of libido in men, and breast tenderness or menstrual irregularities in women. Inhibitors of cytochrome 3A4, that is CYP3A4, example clarithromycin, itraconazole, etc., increase its blood levels, while inducers like carbamazepine, rifampin, etc. may decrease its efficacy. Both trimetrin and amiloride are used in conjunction with a thiazide type or a high ceiling diuretic to prevent hypokalemia and slightly augment the natriuretic response. The antihypertensive action of thiazide is also supplemented. Risk of hyperkalemia is the most important adverse effect of amiloride and trimetrin. These drugs should not be given with potassium ion supplements. Dangerous hyperkalemia may develop. Hyperkalemia is also more likely in patients receiving ACE inhibitors and ARBs, beta blockers, NSAIDs, and in those with renal impairment. Both drugs elevate plasma digoxin levels. Module 4. Drug-drug interaction with reference to antibiotics. Antibiotics are substances produced by microorganisms which selectively suppress the growth or kill other microorganisms at very low concentrations. This definition excludes other natural substances which also inhibit microorganisms what are produced by higher forms like antibodies, or even those produced by microbes but are needed in high concentrations like ethanol, lactic acid, H2O2. Increased incidence and variety of adverse effects. Toxicity of one agent may be enhanced by another. Example, vancomycin plus tobramycin and gentamicin plus cephalothin produce exaggerated kidney failure. Plasma concentration of theophylline, caffeine and warfarin is increased by ciprofloxacin, also by norfloxacin and pfloxacin due to inhibition of metabolism. CNS toxicity can occur by concurrent use of theophylline and fluoroquinolones, that is FQs, or NSAIDs may enhance the CNS toxicity of FQs. Seizures are also reported. Antacids, sucralfate and iron salts given concurrently reduce absorption of FQs. Ciprofloxacin. It is the most potent first generation fluoroquinolone active against a broad range of bacteria. The most susceptible ones are the aerobic gram-negative bacilli, especially the Enterobacteriaceae and Neisseria. Theophylline, warfarin, cyclosporin and zidovudine pharmacokinetics has been found to remain unchanged during levofloxacin treatment. Beta-lactam antibiotics. These are antibiotics having a beta-lactam ring. The two major groups are penicillins and cephalosporins. Penicillin was the first antibiotic to be used clinically in 1941. Hydrocortisone inactivates ampicillin if mixed in the in vivo solution. By inhibiting colonic flora, it may interfere with deconjugation and enterohepatic cycling of oral contraceptives, which results in failure of oral contraception. Probenecid retards renal excretion of ampicillin. All cephalosporins are bactericidal and have the same mechanism of action as penicillin, that is, inhibition of bacterial cell wall synthesis. Tetracyclines. These are a class of antibiotics having a nucleus of 4 cyclic rings. Enzyme inducers like phenobarbital and phenytoin enhance metabolism and shorten the half-life of doxycycline. Chloramphenicol inhibits metabolism of tolbutamide, chloropropamide, warfarin, cyclophosphamide and phenytoin. Toxicity can occur if dose adjustment are not done. Phenobarbital, phenytoin, rifampin enhance chloramphenicol metabolism and reduce its concentration. Hence, failure of therapy may occur. Being bacteriostatic, chloramphenicol can antagonize the cidal action of beta-lactams, aminoglycosides on certain bacteria. Avoid concurrent use of other nephrotoxic drugs like NSAID, amphotericin B, vancomycin, cyclosporin and cisplatin. Cautious use of other potentially ototoxic drugs like vancomycin, minocycline and furosemide through clinical evidence of potentiated ototoxicity. Cautious use in patients more than 60 years age and in those with kidney damage. Cautious use of muscle relaxant in patients receiving an aminoglycoside. Do not mix aminoglycoside with an drug in the same syringe or infusion bottle. Aminoglycoside antibiotics. These are a group of natural and semi-synthetic antibiotics having polybasic amino groups linked glycosidically to 2 or more amino sugars, that is streptidine, 2-deoxystreptidine, garosamine residues. Streptomycin was the first member discovered in 1944 by Boxman and his colleagues. Macrolide antibiotics. These are antibiotics having a macrocyclic lactone ring with attached sugars. Erythromycin is the first member discovered in the 1950s. Roxithromycin, clarithromycin and azithromycin are the later additions. Erythromycin inhibits hepatic oxidation of many drugs. The clinically significant interactions are rise in plasma levels of theophylline, carbamazepine, valproate, ergotamine and warfarin. Several cases of QT prolongation, serious ventricular arrhythmia and death have been reported due to inhibition of CYP3A4 by erythromycin, clarithromycin, resulting in high blood levels of concurrently administered terfenadine, estimazole, cispramide. In conclusion, I can say that most potential drug interactions can be recognized by applying principles of clinical pharmacology and good clinical care. Increased vigilance by clinician at the time of changing drugs improves the chances of identifying unwanted drug interactions before they cause significant harm. With all these information here, we come to the end of today's lecture. Do keep in mind what we have discussed today. This is time for your self-study. If you want to learn more of enhancing your knowledge, you may log on to our website, that is www.cec.nic.in for MCQ, quiz and LORs. Till then, keep studying. Goodbye.